we've talked a lot about some of the proven targeted therapies for subsets of patients. There are some additional subsets like HER2 mutations, and more recently, a lot of excitement about the detection of MET mutations, MET exon 14 mutations. I want to quickly get your thoughts on how do you approach HER2 mutation first and then MET mutation. Perhaps I start with you, Marina, about HER2 mutations.
So for a HER2 mutation, we put the patients into the clinical trial. We know that HER2 is not the HER2 of breast cancer because we have the mutation in the HER2 is not the amplification by FISH, where the results are scanty and that they are not very clear. For HER2 mutation, we have a clinical trial and we include the patients in the clinical trial. I think that the HER2 mutation is just a bit different from the other driver mutations, and the story is quite complicated. While the story of MET seems more clearer, and it is also important to underline that maybe in this case we have a phenotype because the MET14 is present in adenosquamous and in sarcomatoid tumors, and if you have this phenotype and you search for this, you can find it. And the MET inhibitors, also the clinical trial with the MET inhibitors, but also crizotinib are really active in this kind of mutation, and the progression-free survival is long, is about 10 months. And we know also that this kind of driver mutation maybe are not the best candidate for immunotherapy because the results on them are quite poor. So I think that we can target them. The story of HER2 is less clear than MET, and the story of MET is evolving very rapidly, and we have the phenotype to search it.
So there were 2 studies reported at ASCO looking at specific HER2 inhibitors. The activity was relatively modest, as I recall. Benjamin, what are your thoughts on that?
No, I agree. MET is really like the other target, so it's a good target, a good drug, and it's 10 months PFS, so it's homogeneous. But HER2 story is more complicated. It's clear that the tyrosine kinase inhibitors in HER2 mutated NSCLC are poorly active. It's probably the antibody against HER2 that are more active. TDM1 is not that active, but there are, let's say, next generation antibody drug conjugate against HER2, and we should see the result with this drug. There is also the combo trastuzumab, pertuzumab that is used in breast HER2 positive cancer that could be tested, because we feel that there is, adding an antibody to a chemo, an HER2 antibody to a chemo, brings something in these patients. There is retrospective data that strongly suggests that, but we would need prospective data in this population.
Giorgio, your experience with the MET inhibitors that you've seen evolving?
Well, I truly believe that the only area in which the MET inhibitors reasonably will be active is in the field of the c-MET amplification. So again, MET is a target. MET is an important target. There are preclinical data showing that the MET inhibition is relevant when you have a c-MET amplification in the cell line. And it's also true in the initial clinical evidence that we got across several MET inhibitors, at least in the early phases of the clinical development. It's a matter of fact that there is a converging line of research across several institutions and pharma companies that are looking specifically to c-MET amplified tumors, because the very preliminary data that we got are talking about, even in the context of a second line, about 25–27% response rate. Obviously, we don't have any data on PFS, but that is at least a ground in which you can explore, because I do not believe that the c-MET inhibition is a kind of inhibition that should be investigated in all comers.
So at this point, then it's fair to say for HER2, MET, the targeted options are still evolving. They should still get platinum-based chemotherapy for sure as first line, and then subsequently, if there's appropriate clinical trial to test these inhibitors, then we would highly encourage those patients to participate in this clinical trial.
Absolutely, yes.